Hi, Ecology Lab. This week's lab exercise is sort of a do-at-home lab. I'm going to walk you through that a little bit. But the main content, the main topic of the lab is the concept of ecological niche. And niche is kind of a complicated concept, but in general, it's basically the role that a species plays in its ecosystem. So it's the resources that it requires and the resources that it uses. It's how it influences its environment by changing resources and how it interacts with other species. So we can often think about the space in an ecosystem that a species occupies is its niche. The word derives from niche or niche, which is an architectural term. If you see over here, this statue in this little carved out area, that's called a niche or a niche, because the little concave area in the wall is fit exactly to the size of the statue. So you can think of the statue as being the organism or the species, and the niche as being the sort of place it fills. You can see down here in these graphs in the lower left, a species niche, we can map it on sort of different axes, right? So if there's resource 1 on the x-axis and resource 2 on the y-axis, you know, the amount and the types of resources that the species uses. So if those are some kind of habitat or things it eats or space it takes up or energy it obtains from somewhere, we can map those out. Furthermore, over here in the second graph, we have the concept of realized niche, which is when multiple species interact, competition can often make those niches smaller. So species 1 it gets outcompeted by species 2 and species 3, and so now the niche that it actually occupies in the ecosystem is much smaller. Of course, facilitation can also make niches bigger. Okay, so how do we measure niches, and what are sort of meaningful ways that species occupy niches? Well, there's any number of ways that species can, that we can measure niches, because species have, many different kinds of requirements and play many different roles in an ecosystem. This graph in the middle of the slide here is a multi-dimensional graph, an ordination graph, which we'll probably talk about later in the course. But basically what it's showing is that there's lots of different axes that a species niche can be measured along. And so we're only really good at seeing graphs in sort of 2 or 3 dimensions. What this plot is trying to show is that we've got many dimensions which we can't really visualize. But there's lots of ways you can measure niche space. However, the most sort of agreed upon measures of meaningful niches have to do with microhabitat and diet. So basically, where does a species live and where does it forage? Where does it, do its stuff on a very small spatial scale? And then what does it eat and how does it obtain its food? Those tend to separate species into different niches. Okay, so a really classic study on niches was done back in the 50s by this guy, Robert MacArthur. Robert MacArthur was an early ecologist, a really famous one, did a lot of really influential work in ecology. And basically, he was working on the coast of Maine, and he was looking at these 5 different species of warblers, of wood warblers. You can see them here: the myrtle warbler, Cape May warbler, Blackburnian, black-throated green, and bay-breasted. And what he noticed is these birds all seem to occupy the same niche. They're about the same size. They have a very similar diet. They all eat mosquitoes. They live and nest in the same places. In fact, they're foraging around the same species of tree. They're foraging in balsam fir. So MacArthur wondered, how can all these species coexist? Why wouldn't one or a couple of them outcompete the others and drive them either to extinction or drive them out of this habitat? And so what he did is he observed the feeding patterns of the different species. He spent hours and hours watching where the birds are foraging for insects. And what he found is that if you take a balsam fir, a tree, and you divide it into these different zones, right, from top to bottom and from sort of the outer tips of the branches towards the inner part of the canopy, the different species spent different amounts of time foraging in different parts of the canopy. And so MacArthur argued that this is a way of partitioning that niche space, right? Partitioning the niche so that the species don't compete directly. So what you're going to do in this lab is we're going to do a similar thing to what MacArthur did, but on a a little more basic level, in that we're just going to watch the foraging of birds at bird feeders. So this is the setup of the feeders at the Cornell Lab of Ornithology in Ithaca, New York. And it's a really heavily visited feeder setup. There's lots of birds and lots of different species. And the feeders are different designs, right? So there's different kinds of feeders, so the birds that might forage different ways. So our big question here is, do the different types of feeders really represent niches for the birds? Are they going to use some over others and sort of partition their niche space? So if you count these, I think there's 6 feeders here. So there's that sort of flat platform feeder at the bottom. Over here on the left, we've got some kind of cylinder feeder. Looks like it's made out of wood and bark. Up here, we've got a suet feeder. You can't see this very well, but we're looking at the side, but that's a cage feeder. So there's cages on both sides, and then there's suet in there, so birds can kind of climb on the sides of the cage and then peck through. Over here we have another different kind of suet feeder. This one's almost empty in this image. And then we have this tube feeder with perches. So what you're going to do is kind of like MacArthur did. You're going to observe which species and how often they visit the different niches. So you'll go to this website at the top, and that'll get you to the live camera that's pointed on those feeders all the time. If you scroll down that page, there's some information on the most common birds that you'll see there. So if you need help identifying the birds, you should scroll down and study those. You can click on those species and get more descriptions and more pictures of them. These are the most common. There might be a couple others as well. So what you'll do to collect your data in this lab is you'll watch that live camera for an hour. You probably want to watch it when it's morning or evening in Ithaca to see the most birds. And so what you're going to do is identify the species of birds that you see, and then make a note every time a bird of that species visits the different feeders. So I've numbered my feeders here, and then I have so for instance here we can see there's 2 mourning doves on the platform feeder, so I put a 2 on feeder number 1. Back here there's a hairy woodpecker on the suet feeder. I've numbered that one feeder number 3. There's a red-winged blackbird right here on the tube feeder, which I've labeled number 6. So what you might want to do is just put this down in a notebook and make tick marks and count the numbers of visits by different species at the different feeders. Then to analyze your data, we're going to use a statistic called chi-square, which you might have seen before in genetics. And what chi-square does is it tells us if the observations that we see are different from a random expectation, or at least an expectation where everything is evenly distributed. And we have categorical data as both our dependent and independent variables here, right? So our niches are categories, right? The different feeders, 1, 2, 3, 4, 5. And the species are different categories too, right? Blue jay, goldfinch, whatever. So we need to use a chi-square here to see if our pattern is different from sort of random. So the chi-square statistic tells us if the pattern we observe is different from the expectation where birds are evenly distributed. So if we just divide the total number of visits by the number of niches, then we would expect that to be sort of our random or our null expectation. And if our observed is significantly different, then we can conclude that these niches are actually partitioning bird foraging. So in the equation above, O is the observed number of visits, so you'll take your data and plug it into this equation. E is the expected number, so if all visits were the same. So in the lab, there's a lab packet. There's data tables to guide you through calculating chi-square. So just follow those steps. You'll plug your data in, work through this equation. Later on in the semester, we're going to use a different approach, a little more powerful approach to sort of address the same question. Is behavior random or not? And we're going to use a null model simulation instead. But right now, for this lab, we'll do the chi-square. So observe those birds and take your data and then run it through the analysis. This will be due next week. So you may ask yourself, well, why should I care about ecological niches? What is the point of doing this chi-square analysis and seeing if things are random or whatever? Niches are really, really important to understanding ecology. In fact, for applying ecological principles to a number of really important conservation challenges. So number one, the number one cause of species extinction and species loss and loss of biodiversity is habitat loss, a human-induced habitat loss. So we do a lot of land clearing and conversion of one type of land to another, forest to agricultural land, grassland to agricultural land. This is an image of deforestation in the tropics. And so when we do that, we're going to disrupt niches, we're going to disrupt species, and that's why we're losing biodiversity so rapidly. So if we can understand species niches and we can make better decisions about what to conserve, what kinds of habitat to conserve, and so on, if we want to protect certain species or if we want to protect biodiversity. Another reason we should care about niches is invasive species, which again further reduces biodiversity. And so for instance, here on the left, we've got an eastern bluebird. And bluebirds are a native species to North America, and they're cavity nesters. So one of their components of their niche is that they raise their young in cavities. Now on the right, that's a European starling. Now starlings were introduced by humans into North America, and they very rapidly spread across the whole continent. And they're actually outcompeting eastern bluebirds where their ranges overlap, and because they're cavity nesters too. And starlings are actually very aggressive and they can kill eastern bluebird chicks if they're in a cavity that the starling wants to nest in. They can even kill adult bluebirds. So it's leading to a species decline. But understanding the niches of lots of species, when you have introductions, you can understand the impacts that they might have on their non-native ecosystem. And so that really helps with the management of invasive species, which again leads to invasive species can lead to biodiversity loss. And finally, another reason that understanding niches conceptually and practically is important is because of climate change. So human-caused climate change is causing a whole lot of disruption to biota. And so if we can understand the niche space that species occupy, understand the environmental constraints, understand their interactions with other species, then as we can model climate forward and see what changes are likely, we can see how that's going to affect species ranges. And that's, again, really important for things like if we want to conserve biodiversity, we may have to assist species migrations, right? If climate change is too rapid for species to migrate, we may have to move species to suitable habitats. How do we find out what suitable habitats are? Well, we have to understand their niche space. We have to understand the controls on those species distributions. So understanding niches and the tools to do it is not some just purely academic exercise. It has real applications in the real world that's important for biological conservation.